Here we will study mitosis. What is mitosis? What is karyokinesis, cytokinesis, centrosome, microtubules, and at the end we will discuss its importance as well. First of all, what is mitosis? Mitosis is the division of somatic cells. Well, mitosis is the division of parent cell into two daughter cells having same number of chromosomes like that in parent cell. It can happen in haploid cell as well as in diploid cell, and it almost happens in nearly all parts of the body if and when required. So this mitosis is divided into two further phases, which are karyokinesis and cytokinesis. What happens in karyokinesis? That the centrioles, these are the centrosomes having a pair of centriole in it. This is a centrosome and these are the centriole, two centrioles inside of it. So these are formed during interphase, which is a phase before mitosis. So these replicate during interphase, and now in mitosis what happened, these centrosome will move toward the opposite poles of the cell. Like you can see, this will come there and the other will move toward the other pole. So in karyokinesis, first you should know what do you mean by karyokinesis. Karyo means nucleus and kinesis means division. Here you can see the division of the nucleus occur. Each cell has its own nucleus. And in cytokinesis, cyto means cytoplasm and kinesis means division. So here cytoplasm will be divided. Each cell will have its own cytoplasmic region. Here the cytoplasm is shared between two cells. Okay, so this is just the division of the nucleus, but here the division of cytoplasm happened. And this, so we will see this in detail. You can see this is the centrosome. In a cell, the centrosome is present at the pole. So this is the centrosome, and centrosome is consist of two centriole, first and the second one centriole. Centriole is a cylindrical shaped organelle. Its function is to form microtubules. Microtubules are composed of tubulin proteins and some part of RNA. Here you can see that this is the centriole. First it formed three kinds of microtubule. First one is estral microtubule, second one is kinetochore microtubule, third one are the polar microtubule. Okay. So estral microtubules, here its function is to remain, these are the estral microtubules. Its function is to attach the spindle to the cell membrane because there the cell is present. Okay, this like this. So it's these are basically the estral microtubules which help to remain the spindle attached to the cell membrane. Next one is the kinetochore microtubule. These are the kinetochore microtubules which are attached to the central region of the chromosome, and this central region is what we call kinetochore. So these spindle fibers are attached to the kinetochoric region. That's why we call kinetochore microtubules. So third one are the polar microtubules. These polar microtubules help to separate this sister chromatid, which will happen later in mitosis. These sister chromatids will be separated. Like this is a chromosome, so it would be separated. What happens? That these sister chromatids would be separated. Where these polar microtubules. Here you can see this is a chromosome. It consists of two sister chromatids, and this is the centromeric region. The central part of the chromosome is called centromeric region, and here the centromeric region have an area called it is called kinetochoric region. Okay, where the spindle fibers from the centrosome would be attached, which we call kinetochore microtubules. Here you can see that this is the centrosome. It consists of two centrioles. Here the spindle fiber, the estral spindle fibers are coming. This is the kinetochore spindle fiber, and these are the polar microtubules or spindle fibers. So here we can see the first phase of mitosis, which we call prophase. In prophase, the chromosomes are condensed. Before this prophase, there is the phase which we call interphase, where the chromosome appear as the chromatin. Which this is a DNA, okay, where the DNA is invisible. Even it is here we are unable to see it with the electron microscope. We have to use the histologic stain to see this chromatin material, to see this DNA material. But when the cell enters in prophase, in this phase the DNA will be condensed, the DNA will be thick and visible. It becomes short and visible. You can see that the sister chromatids will become attached to each other and form a chromosome which would be visible. And in this phase, nuclear membrane, it would be disintegrated. You can see it would be disintegrate and nucleus will disappear in this phase. When the nuclear membrane disintegrate, the nuclear material would released into this cytoplasm, so that further cell division process continues. So the cell next the cell would be entered into the metaphase. Here the chromosomes arrange themselves at the equatorial plate. You can see we are using spindle fibers. These are the spindle fibers. And in metaphase, the spindle fibers are attached to the kinetochoric region of the chromosomes. So next phase is the anaphase, where the chromosomes would be pulled apart. So here you can see that the spindle fibers would be contract and pulling these chromatids towards the opposite poles, like this. So they are basically pulling these chromatids toward the opposite poles of the cell. So this is the cleavage furrow cytokinesis. This is a contractile ring here is forming. This is because of the spindle fibers which are pulling these sister chromatids toward the opposite poles, and then the cell would be divided into two daughter cells. Here in telophase, we can see that chromosomes are becoming uncoiled, they are becoming unfolded, due to which they would appear as chromatin, because this, you can see these DNA would become invisible now, and the nuclear membrane starts reforming. Here the nucleus will also reform, because the cell is dividing into two. So whatever the cell needs, like it needs the centriole, so centriole will also be present in both the cells, and nuclear membrane would be reformed, and nucleus will reappear, and the chromosome, they will become uncoiled and will appear as a chromatin. So here the telophase ends. So this is the overview of the mitosis. The first one is the prophase, in which the chromosome become condensed and nuclear membrane would be disintegrated and nucleus would be disappeared. In metaphase, the equatorial plate is formed, where the chromosomes are arranged to form the metaphase plate. And the next one is anaphase, in which spindle fibers are pulling the sister chromatids toward the opposite poles. In telophase, what is happening there, that the cell is dividing into two daughter cells having same number of chromosomes like it in parent cell. Last is the importance of mitosis. Mitosis is very important because like in order to regenerate the cells, for even cells growth, or to replace the older cells, we use mitosis. So all these processes happen through the mitosis. So if it should be regulated wisely, it means if there is any mistake in mitosis, it would result in lethal diseases like cancer or tumor or malfunctioning. So that's all about mitosis. If you like this video, then please like and subscribe our channel so that we would be able to make new videos like this.